All right, sorry about that, guys. Um, my phone started ringing and it was a phone call that I had to take, so I apologize. I'm not able to pause these recordings. So I just had to end the first one, and now I will pick up where we left off. So we're talking about unsaturated fatty acids and how unsaturated fatty acids are characterized by the existence of at least one double bond between carbons in that fatty acid side chain. Now, because these carbons have a double bond, that double bond translates into sort of a kink right here in those side chains. So, depending on where the double bond is, that's where the fatty acid will have a little bend to it. And because now we have these triglycerides with sort of these bent fingers coming off of the side, because those fatty acids are bent, it makes it difficult for those triglycerides to pack together tightly, like we saw with saturated fats. So because these can't pack together as tightly, we have unsaturated fats that exist as liquid at room temperature. All right, so we're talking about vegetable oils. You'll notice there, if you go to the grocery store, you see them at home, they're going to be a liquid in that bottle. And that's because they're made up of unsaturated fats that aren't able to pack together as tightly. Unsaturated fatty acids, a couple of different types here. Mono unsaturated fatty acids, mono being the key prefix there. Mono means one. So, as I'm sure you can guess, a mono unsaturated fatty acid is only going to contain one double bond along the carbon chain. All right, so in this example, that one double bond is right here. Then we have polyunsaturated fatty acids, and poly, that prefix, means many. So polyunsaturated fatty acids are going to contain two or more double bonds along the carbon chain. Now in this example we have three, one right here, and one right here. So this example on the bottom would be a polyunsaturated fatty acid. Unsaturated fatty acids bend at the double bond site, which we just chatted about, and that changes the shape of the fatty acids. This means that they are not able to stack like saturated fatty acids, therefore, they manifest themselves as liquids at room temperature. Now, not only does the number of double bonds matter in an unsaturated fatty acid, but also the location of the double bond. So, in polyunsaturated fatty acids, remember those are the fatty acids with two or more double bonds. In polyunsaturated fatty acids, chemists identify the location of the first double bond from the methyl or omega end by assigning it the Greek letter omega. And that's the Greek letter right there. It looks kind of like a W. That's called an omega. So what we're talking about, and you'll see an example on the next slide, is we're counting in the carbons from that free end of the fatty acids. And when you count in 3 or 6 fatty acids, that is what designates a fatty acid as an omega-3 or an omega-6. And again, both omega-3 and omega-6 fatty acids are considered polyunsaturated. And their names simply come from the location of the first double bond on the carbon chain. So here we have them in this example. Both omega-3 and omega-6 fatty acids are considered essential. So what does it mean to be an essential fatty acid? Well, it means that your body doesn't have the ability to synthesize it on its own, which means you can't make your own omega-3 and omega-6 fatty acids. You have to get them from your diet. So it's very, very important to make sure that you're eating foods that do contain omega-3 and omega-6. Other names for omega-3 is linoleic acid, and omega-6 is also referred to as alpha-linolenic acid. And you can see here what we're talking about. So this blue end right here, this is the free-floating end. This is the methyl or the omega group. This end over here on the right is the acid group. This carbon double bonded to an oxygen with a hydroxyl group here. This is where the fatty acid molecules are bonded to the glycerol backbone. And so, as I said before, both the omega-3 and the omega-6 are considered polyunsaturated, meaning they have 2 or more double bonds. So the alpha-linolenic acid is the omega-3, and we see that down here at the bottom. This double bond here, 1, 2, 3 carbons in from this free end. That's how we know that that's an omega-3. The linoleic acid, 1, 2, 3, 4, 5, 6. Count 6 carbons in from the methyl or omega end, and that is going to designate this fatty acid as an omega-6. Omega-6 fatty acids, again, linoleic acid is used to make arachidonic acid or ARA, which is another type of omega-6 fatty acid. The human body does not have the enzyme to make omega-6s and therefore can only make arachidonic acid if linoleic acid is provided from the diet. Again, what we were talking about before about omega-6 fatty acids being essential. Different dietary sources of omega-6s are corn oil, safflower oil, soybean oil, and nuts. Omega-3 fatty acids, also referred to as alpha-linolenic acid or ALA, can be used to make eicosapentaenoic acid, or EPA. That's a lot easier. And we'll just say DHA. So you've probably actually heard of EPA and DHA before. They're usually just referred to by their abbreviations, and I know I've seen them in commercials when they talk about sources of omega-3 or different multivitamins, things like that. EPA and DHA are also types of omega-3 fatty acids. The human body, again, does not have the enzyme to make omega-3s and therefore can only make EPA and DHA if linoleic acid, that should be alpha-linolenic acid, is provided in the diet. Dietary sources of omega-3 include flax seeds and flaxseed oil, fish, walnuts, and grass-fed or pasture-raised animals. Omega-3 and omega-6 ratios. The U.S. diet contains a ratio of omega-6 to omega-3 that is less than optimal for health. The recommendation is 15 to 1, omega-6 to omega-3, and the U.S. average is about 15 or 20 to 1. Excuse me. Reducing the consumption of processed vegetable oils such as soybean, safflower, corn, and canola oils, and consuming more fish and flaxseed oil can help to correct that ratio. Mixed triglycerides, the fat that we eat, for example, triglycerides, is usually a mix of unsaturated and saturated fatty acids. So typically, you're not just consuming a wad of unsaturated fat or a wad of saturated fat. Typically, there's a percentage of both in foods that we eat. Liquid oils also contain some saturated fatty acids within the triglycerides. They just contain a larger number of unsaturated than saturated. Solid fats also do contain some unsaturated fatty acids. However, the number of saturated fatty acids is far greater than unsaturated fatty acids. This photo comes from your text. It's just an example of what a mixed triglyceride might look like. So triglyceride, we still have these 3 fatty acids and the glycerol backbone, but in this example, one of these fatty acids is saturated. You can tell because it's straight, and the other 2 are unsaturated because they have this bend in them. So this would be an example of a mixed triglyceride because it contains 2 different types of fatty acids within one molecule. This graph comes right from your text, and it's super, super interesting, I think. You're not going to have any of this memorized for the exam, but I like to keep it in here because there's a lot here that we don't necessarily think about. So, as we go on talking about health and fats, we're going to talk about some of the health risks of a diet high in saturated fat and low in unsaturated fat. And I think this is particularly important right now as we've really seen this huge surge of popularity around coconut oil. I am not a fan of coconut oil. And that's really just because of how unhealthy it is. Um, some people like the taste of it, that's fine. But it's really getting promoted as sort of a healthy alternative to fats like butter, and that's really just not the case at all. So, what you'll see here on this graph, down the left hand side here, we have all different, foods, sources of fat. And the different colors on these bars show us what types of fat are in each of those foods. So you'll notice way down here, flaxseed. Of all the fat that's in flaxseed, a tiny bit of it is saturated. A little bit more of it is monounsaturated, but then the vast majority of fat in flaxseed comes from omega 3 and omega 6 polyunsaturated fats. So that's great. All of this is unsaturated. So, very, very good. We want to limit saturated fat in our diet. Same thing, all these oils, they're liquids at room temperature. They have some saturated fat, but not a lot, not too bad. Okay. Then we get up here to coconut oil. Look at this. See all this purple? Coconut oil has more saturated fat than cheddar cheese. It has more saturated fat than butter, and it has more saturated fat than roast beef. Now, if you've seen coconut oil in the store or you have some at home, it's solid at room temperature. Well, that's because it's got so much saturated fat in it. In fact, it has way more than butter. So if you're somebody that's trying to reduce your intake of saturated fat, which we all should, you would be a lot better off just eating regular butter than coconut oil because of all of this saturated fat. Granted, we still have canola oil, olive oil, all of these foods down here have less saturated fat than butter. But if your choice is between butter or coconut oil, I would go for butter every single time. Way healthier for you in terms of the types of fat that it contains. So just something to keep in mind, when you're making decisions about what type of fat to use when you're cooking. Obviously, if you like coconut oil, you like the taste of it, fine. But again, use it in moderation. It's not this super healthy cure-all type thing because it's really just not that good for you and it's pretty expensive. So just some for your information, you can read more about this in your book. Tell your friends, spread the word. Coconut oil is just not all it's cracked up to be. Rancidity. Fats become rancid when exposed to oxygen. This is what caused fats to kind of go bad in a way. Saturated fats are the most chemically stable and therefore do not become rancid as quickly as unsaturated fats, which are more vulnerable due to their double bonds. So something like butter is not going to go bad as quickly as something like olive oil. Hydrogenation. Hydrogenation is something that's been happening for a little while now, but in more recent years, and it's a type of processing that companies have been doing to fat. Excuse me. So in order to protect fats from becoming rancid and to increase the shelf life of foods containing fats, food manufacturers use a process known as hydrogenation. Hydrogenation, the definition, is a process in which hydrogen atoms are added to the carbon-carbon double bonds of unsaturated fatty acids, making them more more saturated. So, hydrogenated oils is what they're called. So they are, oils. They're derived from unsaturated fatty acids. But don't let that fool you, because the hydrogenation process adds those extra hydrogens back in on that fatty acid molecule, causing those unsaturated fats to behave a lot more like saturated fats in your body. So they are not healthy at all. Now, hydrogenation, you might have heard of hydrogenated fats, but once those extra hydrogen atoms are added, they actually become what we know as trans fatty acids. So during the process of hydrogenation, trans fatty acids are created. Now, trans fatty acids more recently have actually been banned in many in food manufacturing. I'm actually not sure if they're allowed anywhere anymore, but trans fats, you would find them a lot in, say, like dessert snacks, like Little Debbie kind of snack cakes, those things that seem perpetually moist when you take them out of the package and you think, God, how can this thing stay so moist in this box on this shelf for months and months and months? Well, it's because it contains hydrogenated fat. So it allows them to fill that snack up with all kinds of fat without it going bad on the shelf, because it's full of trans fats. But now that has been banned, as I said, in most, if not all, foods, because it is really not healthy for the body. So this photo down here at the bottom is from your text, showing a visual representation of the difference between the different types of fatty acids. So over here on the left, we have what we would consider to just be a normal unsaturated fatty acid. We have the double bond here between these two carbons. When hydrogenation occurs, the hydrogens on these two carbons with the double bonds are swapped, so that instead of occurring on the same side of the double bond, now they are on opposite sides of the double bond. So you're like, okay, yeah, what's the big deal? Well, it is a big deal, because when those two hydrogen atoms are moved so that they're on opposite sides of the double bond, it allows that fatty acid to be straight. And when that fatty acid becomes straight, even though it has a double bond, it's straight. And when it's straight, it packs together. It acts as a saturated fat, and your body treats it like a saturated fat, which is really bad for your body. So you might be, probably you're not kind of a science nut like I am, but I always thought it was fascinating, just the one movement of a hydrogen molecule a hydrogen atom from one side of the molecule to the other can really cause such a disruption in how a molecule operates and how your body responds to that molecule. So that's the process of hydrogenation, just taking an otherwise healthy unsaturated fat, moving that hydrogen atom, causing it to become a trans fat, right? This is an example of a nutrition facts label, and actually nutrition facts labels are going to start looking a little bit differently. They just recently, within the last year, started making some changes to kind of make different parts of this stand out a little bit more. But traditionally, you can see trans fats listed right here at the top underneath total fat. And then another good indication would be down here in the ingredients label. If you're reading the ingredients and you see something like cocoa butter or soybean oil, partially hydrogenated cottonseed oil, that word hydrogenated, that's going to be a key that there's trans fat in it. Now, in this example, it says 0 trans fat, but yet down here in the ingredients, it says partially hydrogenated cottonseed oil. Well, according to law, if a product has less than 0.5 grams of trans fat per serving, the nutrition facts panel can list the amount of trans fat as 0, even if partially hydrogenated oil is an ingredient. So what does this mean? Well, you might eat these chocolate chip cookies thinking that you're not going to be consuming any trans fat, when actuality there is a little bit. There's like a half a gram or less of trans fat in this product. But if you look up here, the serving size is 3 cookies. 3 cookies. I don't know what kind of cookies these are. These might be really small cookies. Maybe you sit down and eat 6 cookies or 9 cookies, and suddenly you're eating multiple servings of these cookies. So instead of only having what would be considered a negligible amount of trans fat, that isn't even listed here, because you're eating multiple servings, you actually are eating maybe 1 gram or 1.5 grams, amounts that would ordinarily be listed, but because the serving size is so small, they don't have to list it. So that's kind of a sneaky way that manufacturers can get around actually listing trans fat in the nutrition facts label. But they do have to include it down here in the ingredients. So it's always important to look at the ingredients label to make sure you're getting the full story. Trans fat in the news, the whole margarine versus butter debate, trans fat added to nutrition facts panel in 2006, and some trans fat bans. There was, I don't remember if it actually went through or not in New York City, but I think nationwide now there's been a real crackdown on trans fats, and I don't know that they're really in many foods anymore. Not sure. Trans fat versus palm oil. This is another palm oil you see a lot. Trans fat food labeling has led to food producers to replace hydrogenated oil with palm oil. And so you don't see that word hydrogenated, so you think, okay, it's not hydrogenated, it's not a trans fat, not bad. Wrong. Palm oil is a tropical oil that's very high in saturated fat. And in order to, this increased use of palm oil has actually led to rainforest destruction across the globe. So both in terms of environmental consequences and health consequences, if you see palm oil in the ingredients of a food, it's best to really steer clear or limit your consumption of that food item. All right. This is a breakdown of sort of what we've talked about so far in terms of we have triglycerides broken down into fatty acids and a glycerol backbone. Those fatty acids can be saturated or unsaturated. If they're unsaturated, they can be mono or poly, and if they're poly, they might be omega-3 or omega-6. So I'm not going to spend a lot of time here, but again, this is just sort of a graphic representation of the breakdown to kind of help you understand the connection between all of the different topics we've talked about to this point. Good sources of fat. Coconut oil really should not be here. I need to go ahead and delete that. But tree nuts, avocados, walnut oil, flax seeds and flaxseed oils, grass-fed beef and dairy, wild-caught fish, and pasture-raised eggs. All right, so we talked about triglycerides. 2 left, phospholipids and sterols. So next up, phospholipids. All right, phospholipids. This is what a phospholipid molecule looks like. You might notice, looks a little different than a triglyceride. All right, we still have fatty acids, but here we only have 2. And we still have this glycerol backbone, but you'll notice this bulge here. That's because of the addition of the phosphate group, right? Which you can tell from the name, phospholipids. All right, so there's a phosphate group, big deal. What does that matter? It actually matters a lot. So the addition of this phosphate group to the glycerol backbone makes this green end water soluble. All right, and then these fatty acid tails are fat soluble. So you have fat soluble at one end, water soluble at the other end. That's important, right? So the addition of the phosphate group to the glycerol is the difference here with a phospholipid, and you still have the fatty acid side chains. But water soluble at one end, fat soluble at the other end. That's important to remember moving forward. So because the phospholipid is water soluble at one end and fat soluble at the other end, it has the unique ability to mix with both water and fat. Now, it's present in small amounts, excuse me, in food and in the body. Lecithin is the most common phospholipid. It's made in the liver, and therefore it's non-essential, right? Because your body can produce it on its own, so it doesn't need to get it from food. Lecithin is a major constituent of cell membranes, which we'll see on the next slide. But you can also find lecithin in foods. It should be listed on the ingredients label. It's used as an emulsifier, so it's used to help mix oil and water together. So a good spot to find lecithin, in food would be in salad dressings. All right, so this picture's from your book. This is a cross section of a cell membrane. All right, so we have this animal cell right here. You can see the nucleus, and we've got some mitochondria over here. All right, so we're talking about the cell membrane that kind of holds everything in the cell together and separates it from this aqueous environment out here. So we have the cell interior, this blue area, and that's aqueous. Aqueous means that it's made up of water. Has the same polarity as water. And the cell's exterior is also aqueous. So water outside, water inside. So when we look at the cell membrane, it's made up of a phospholipid bilayer. Bilayer meaning two, like bicycle has two wheels. Bilayer has two layers. One layer on the outside, one layer on the inside. And then so these green heads here, these are the phospholipids. And this is that green head that we saw, that's the glycerol backbone with the phosphate group in it. So this is the water-soluble end of the phospholipid. So you'll see this water-soluble end on both the inside layer against that aqueous environment and along the outside layer, again an aqueous environment. And the fatty acids are going to kind of be extending inward, in towards each other, creating a fat-soluble interior to the membrane. So outside of the membrane, both on the inside here, the surfaces, inside surface and outside surface, are the water-soluble ends, and the inside channel running through the center here, that is the fat-soluble part where the fatty acids are. Okay. All right, now we're going to move on to sterols, which are the third main category of lipids. Ta-da, sterols. So the main sterol that you probably have heard of is cholesterol. Now this is what a molecule of cholesterol looks like. You'll notice it's much different looking than a phospholipid or a triglyceride. Now, instead of having a carbon chain, cholesterol is made up of these carbon rings. So this sort of notation for a molecule doesn't show all of the atoms. Instead, when we're looking at these hexagons here, each of these points, okay, each of these vertices is a carbon. And they just in this way of diagramming a molecule, they don't put carbons, they don't put the C's in, but there's a carbon there, carbon there, carbon there, carbon, carbon, carbon. They only note when the carbons are at the end. This denotes a double bond, and then this, of course, is the OH hydroxyl group. So this is what cholesterol looks like. All sterols, whether they're cholesterol or another type, are going to be made up of these carbon rings. And that is what physically sets these lipids apart from the other two that we talked about. Sterols are in plants. Plant sterols help to form the cell membrane of plants. In animals, sterols take the form of cholesterol. They are made in the liver and therefore are non-essential. Your body can make its own cholesterol. It does all the time. So you don't necessarily have to get it from your diet. Now, when we think about cholesterol, we think, oh, you shouldn't eat very much cholesterol. And that's true because your body can make it on its own. But cholesterol isn't all bad. Having too much is bad, but cholesterol actually has some really important functions in your body. For example, it's used to make cell membranes, myelin sheaths, which are, if you can remember back a million years ago when you were in biology class, the myelin sheaths are sort of like these little cushiony tubes that surround the nephrons of a nerve cell. And when those myelin sheaths aren't present, it can create a lot of issues for your nerve cells. Degradation of myelin sheaths has been seen in things like multiple sclerosis and other issues with the nervous system. Cholesterol is actually one of the components necessary for making vitamin D. It's also important for making bile acids, cortisol, testosterone, and estrogen. And it's important to note that cholesterol is only in animals. I always think it's interesting when I'm in the grocery store and I see a food that is only made up of plants. Like, obviously doesn't contain any animal products at all. So it might be a vegetarian product, anything. And it'll say in big letters on the front, cholesterol-free product. And you think, oh, sweet, no cholesterol. But if it doesn't have any animal products in it, it would never have cholesterol in it to begin with. Cholesterol is only in animal products. You're not going to get cholesterol from eating fruits and vegetables. You're not going to get cholesterol from eating, I don't know, something with only plants in it. So sometimes they use that as a marketing tool, saying something is cholesterol free, but it's not that it's any healthier because it never would have had cholesterol in it to begin with. So just something to keep an eye out for. Dietary cholesterol, sources of cholesterol in our diet, typically beef, pork, chicken, fish, eggs, and dairy. Again, all animal sources. And this is right from your text as well, showing you the largest contributors of cholesterol in our diet. And again, you can see no plant sources, so no surprises there. Lipid digestion, absorption, and transport. This is all stuff that we talked about in your digestion chapter, but I do want to go over it again, specifically with regard to lipids, just to make sure that you all fully understand lipid digestion. Let's see. So here we go. We're going down through the digestive system. Starting in the mouth, no fat digestion, no lipid digestion up here. A very small amount of lipid digestion occurs in the stomach due to the action of gastric lipase. Lipase, we have that ASE suffix telling us that that's an enzyme that, in this case, lipase digests lipids. Gastric lipase produced in the stomach will digest fats a little bit. Then the liver produces bile, which is stored in the gallbladder and released into the small intestine to aid in the digestion and absorption of fat. The pancreas produces the enzyme pancreatic lipase, again, lipase, enzyme that's responsible for digesting fat, and that lipase is released into the small intestine to break down triglycerides into monoglycerides fatty acids, and glycerol. So the lipase is taking that triglyceride and separating those 3 fatty acids from the glycerol backbone. In the small intestine, here again, we have the little brush border. We have the villi and microvilli along the lining of the small intestine. Because your because your blood vessels are aqueous, and fatty acids are obviously not water soluble. Once those fatty acids get broken down, they're going to be packaged into these little bundles called micelles, and there's also, we'll talk about in just a second, different lipoproteins, but those are proteins that help to kind of package fatty acids to allow them to move into the aqueous environment of the body and move throughout the blood. And then since fat absorption in the small intestine is very efficient, we absorb almost all of the fat that we eat, lucky us. Very little fat is actually excreted in your waste. Bile is released from the gallbladder whenever dietary fat is present in the small intestine. Bile acts as an emulsifier. It breaks up big lipid globules into small lipid droplets, which allows the lipases, again, lipid digesting enzymes, to break the triglycerides down to fatty acids and glycerol. The emulsion is similar to shaking a bottle of Italian salad dressing, where you get sort of all those little bubbles of oil mixing with the vinegar. And the existence of bile is necessary for proper fatty acid absorption. All right, as promised, now we're going to chat about lipoproteins. There are 4 different types of lipoproteins. We have chylomicrons, very low density lipoproteins, or VLDL, low density lipoproteins, or LDL, and high density lipoproteins, or HDL. And we're going to go into more detail about each of those. So a lipoprotein structure, we see here again our friends, the phospholipids. They have this water-soluble head on the outside because it's going to be traveling through an aqueous environment. We have these big blue proteins here. It's called a lipoprotein because it's made up of fats and proteins. So again, lipoproteins are like the school bus for fats. So in your digestive system, once fatty acids and cholesterols are all broken down and they need to travel, or just even within your body after they've been absorbed, if cholesterol needs to travel throughout your body, it's going to hitch a ride on this lipoprotein bus, all right? Because cholesterol, triglycerides, they don't like water. They don't like aqueous solutions, and they don't mix very well. So in order to travel through your blood, which is made a lot up of water, they have to get on this lipoprotein structure. So the lipoproteins, the phospholipids here, are going to surround this bundle of cholesterol and triglyceride. And because they have this outer shell of the phosphorus glycerol, they're able to move through the water or through your blood. And the fatty acid tails all kind of stick in here towards the middle, wrapping themselves around the cholesterol and triglyceride so that it can move along through your body. So this is the basic structure of a lipoprotein. There are, as I said, 4 different kinds. We're going to go into more detail on each of those now. First up is the chylomicrons. These are made in the small intestine, and they work to carry large dietary lipids such as long-chain fatty acids, cholesterol, and fat-soluble vitamins through the lymphatic system. Eventually, they enter the bloodstream at the left subclavian vein and then deliver the lipids that it's carrying to the various cells of the body, right? So the chylomicron bus delivers the different fats at the different bus stops, the heart, the muscle, the mammary tissue, and the adipose tissue of your body. All right, one down. Next up, we have the very low density lipoproteins, or VLDL. These are made in the liver, and VLDL carry lipids that were made in the liver and distribute them to the cells of the body. As the VLDL circulate through the bloodstream, they shrink, as they kind of drop off all the fat where they need to go, and some leftover particles do circulate back towards the liver, while others become low density lipoproteins. Low density lipoproteins, or LDL, are derivatives of VLDL. They contain mostly cholesterol. Okay, so you're not going to find a lot of triglycerides on this bus, mostly cholesterol in the LDL. They deliver cholesterol to cells via the blood. LDL is sometimes referred to as the bad cholesterol. LDL isn't actually a cholesterol, but it does carry cholesterol. And the reason why it's called the bad cholesterol is because it delivers cholesterol to the cells, which isn't a bad thing, but it is bad if you have too much. So having high levels of LDL in the blood can mean an over delivery of cholesterol to your cells, which is associated with an increased risk of heart disease. Lipid transport and delivery. This is sort of a more detailed picture of what we just talked about with the different lipoproteins. This isn't anything that you have to have memorized. It's right in your book. I just wanted to include it because it might be helpful if you're somebody that kind of needs to see things a few different ways in order to understand how the different lipoproteins carry fat throughout your body. All right, so 4 different types of lipoproteins. We have chylomicrons. Those are going to carry mostly triglycerides. VLDL also carry mostly triglycerides, but a little bit more of the others. LDL, mostly cholesterol. And then we have HDL. We didn't talk very much about HDL. Where LDL is considered to be sort of the bad cholesterol, HDL is considered to be the good cholesterol. So LDL is a lipoprotein that's responsible for delivering cholesterol to your cells. HDL is responsible for cleaning it up. So where you might have seen like a cross section of an artery, how it's all clogged with fat and stuff. LDL just sort of is dropping off cholesterol in your body. HDL is coming through and sweeping it up, taking up all that extra. So in your body, it's good to have lower levels of LDL because you don't want all these cholesterol deposits, and higher levels of HDL so that if you do have those deposits, they can be cleaned up pretty quickly before they cause a problem. Lipid functions. The benefits of lipids in fats, they taste good. I mean, fat's good. It tastes delicious, provides texture, flavor, aroma to foods. Phospholipids and cholesterol are important structures in cell membranes and also serve as starting materials for other molecules. We'll talk about vitamins in a later chapter, but there are 4 fat-soluble vitamins: vitamin A, D, E, and K. And without fat in your body, you wouldn't be able to absorb those 4 vitamins. So fats are helpful for the absorption of fat-soluble vitamins. In terms of energy storage, fat is lightweight and a good long-term storage of energy. It also provides our bodies with insulation, cushions for our joints, and lubricants for our joints. Storage of excess energy as adipose tissue is, of course, can be seen in fat cells or adipose cells in our body. Lipids and health and disease. This is where we're going to talk about heart disease and some more about that LDL and HDL. More than 80 million people in the United States suffer from cardiovascular disease. It is the number one cause of death. You might have heard the term atherosclerosis. What this is referring to is the hardening of the arteries. All right, so comes right from your text, and this is looking at the cross-section of a normal artery. So here we have the normal artery. Okay, we're having some damage here to the artery wall. LDL, those lipoproteins, they're coming through, they're dropping off some cholesterol. Your white blood cells are coming in to respond to this injury. As we move on, the oxidation from the cholesterol and the injury is going to form a plaque inside of your artery, and the plaque starts getting kind of hard, kind of rigid, and this is what we're talking about with atherosclerosis, all right, the hardening of your arteries. Over time, if that plaque is left unchecked, it's going to grow bigger and bigger and bigger, and as that plaque grows, you'll notice the area inside your blood vessel is getting smaller and smaller. So when that area gets smaller and smaller, it becomes harder and harder for your heart to push blood through that tube, because the tube has gone from being big to being like half the size. And that's when you can have blood vessels rupture. You can have a heart attack because your heart is like really working overtime, or one of these plaques from your blood vessels can actually sometimes become dislodged. And if the plaque falls off the wall of the blood vessel, you might think, oh, well, that's great, plaque's gone. No, the plaque's not gone. The plaque will actually continue moving through your blood vessel until it gets to a smaller blood vessel that it can't fit through, and then it will form a clog. If that clog occurs in your brain, you might have a stroke, and if it occurs elsewhere in your body, you might actually have a heart attack. And that's what we can see here with this plaque rupture. And if the plaque moves through your bloodstream to a smaller blood vessel, that's what we see here, is that blockage in the blood vessel. Risk factors for heart disease: diabetes, high blood pressure, obesity, high blood cholesterol. Age is a risk factor for heart disease. So in men, that's an age over 45, and in women, an age over 55. Gender is a risk factor. Men are at greater risk earlier in life. Also genetics and lifestyle, smoking, exercise, and diet. Obviously, if you smoke, it's going to increase your risk. But if you exercise and eat healthy, it will reduce your risk. Diet and heart disease risk. You are at increased risk if you consume a diet high in sodium, saturated fat, trans fat, and also, eat a lot of red meat. You can decrease your risk by consuming a high fiber diet and adding foods like fish, nuts, whole grains, fruits, and vegetables to your diet. In your diet, you really want to eat foods that will work to decrease your LDL and increase your HDL. This means avoiding trans fat in your diet, also avoiding saturated fat, and increasing dietary polyunsaturated fat and increasing monounsaturated fats. Quitting smoking if you smoke, improving social relationships, reducing stress, and exercising. Eating fiber to reduce blood cholesterol. This is going to connect it a little bit to chapter 4. Diets high in soluble fiber have been shown to reduce blood cholesterol levels. The fiber traps bile in the small intestine and does not allow for the bile to be reabsorbed and recycled. The liver is then forced to pull cholesterol from the blood to make new bile, therefore lowering blood cholesterol levels. We talked about heart disease. Now we're going to just talk briefly about cancer. Cancers are the second leading cause of death. Populations that consume high amounts of fruits and vegetables have lower risks of cancer, whereas populations consuming diets high in fats tend to have higher risks of cancer. Trans fatty acids have been shown to increase the risk of breast cancer, whereas diets high in omega-3 fatty acids from fish reduce cancer risk of the breast, ovary, colon, and upper digestive and respiratory tracts. Dietary fat and obesity. Again, it's going to be important for you guys to know. This is on the test, I promise you. Caloric density. So fat has 9 calories per gram, the highest caloric density of any macronutrient. Carbohydrates and proteins have 4 calories per gram, and not listed here is alcohol, which contains 7 calories per gram. And as we know, lucky us, fat is very efficiently stored and very efficiently absorbed through our small intestine. So if you consume fat, it will likely wind up stored somewhere in your body. Meeting your lipid needs, fat recommendations. It is important to consume fat. In fact, the recommendation is that 20 to 35% of your total calories should come from fat. Linoleic acid, the recommendation is 12 grams a day for women and 17 grams a day for men. Alpha-linolenic acid, the recommendation is 1.1 grams for women and 1.6 grams for men. The recommendation for saturated fat is less than 7% of total calories. The dietary guidelines currently do not include a limit for dietary cholesterol, but because your body is able to make it on its own, they do recommend eating as little as possible. They just don't give a specific number. And trans fats really limit or avoid completely if possible. Healthy MyPlate choices, individual nutrients and food choices, factors that increase risk. Again, saturated fat, trans fat, excess sodium, excess sugar, and excess energy and calories. Factors that reduce risk, polyunsaturated fats, monounsaturated fats, fiber, B vitamins, antioxidants, and a moderate alcohol consumption. Food labels, again, just a reminder of where to find fats on your food labels. This is the same chocolate chip cookie example from earlier in this presentation. Food labels. I promise we're almost done. There's just this slide and one more. You guys are troopers. Food labels, different messaging that you might see on food labels and what it actually means. If a food says that it's fat-free, it has to contain less than a half a gram of fat per serving. Again, just because it's fat-free doesn't mean it doesn't have any fat. It just means that per serving, it has less than 0.5 grams. So another good example of why it's always important to check those ingredients labels to make sure you know what you're actually eating. A reduced fat label, like we see on those Cheez-Its in the back. Reduced fat means that the product contains at least 25% less fat per serving than the regular or reference product. And then a low fat designation, like we see on that Hellmann's mayonnaise in the front. Low fat means that a product contains less than or equal to 3 grams of fat per serving. And again, if you consume multiple servings of that product, your consumption of that fat obviously increases. Last slide. Excuse me, I'm starting to lose my voice here. Fat replacers. There, once upon a time, was an artificial fat called Olestra, which was a sucrose polyester. Sounds great. Olestra was made from sucrose with fatty acids attached. And it can't be digested by either human enzymes or bacterial enzymes in the gastrointestinal tract. Therefore, it's excreted in the feces without being absorbed. Now, you might not have heard of Olestra. It's not really on the market anymore because it was very unpopular. When I was younger, they had a product called O-Chips, And O chips were potato chips that were made with Olestra. And so the idea was that you can eat all the potato chips that you want, but because these chips have Olestra in them, you're not actually going to absorb any of the fat. So you get to taste all that delicious fat without having to worry about it being absorbed in your body. Sounded like a good idea, but if your body's not going to absorb that fat, it has to go somewhere. Which meant that people were consuming these O chips and with all this Olestra, and they weren't absorbing fat, but that meant that they were excreting that fat in their waste. And pooping out fat is not really fun. It's actually very unpleasant. So those O chips didn't last very long, and the use of Olestra didn't last very long. Yeah, ultimately, again, it's just better to eat, high fat, high sugar foods in moderation instead of trying to monkey around with all these different chemical replacers. Ultimately, it usually ends up not being very pleasant. I'm going to go ahead and wrap up here. That is the end of chapter 5. I hope you guys learned a lot, learned a little, learned something. If you have any questions, you know where to find me, and I'll see you guys on Thursday. Bye.